All right, I'll let them all in. Hello, everyone. Welcome to our seminar this week with Dr. Stephanie Colombo. I will start off by reading the land acknowledgement. We respectfully acknowledge that the territory and lands on which Memorial University campuses are situated are in the traditional territories of diverse Indigenous groups, and we acknowledge the histories and culture of Beothuk and the island of Newfoundland as the ancestral homelands of the Mi'kmaq and Beothuk. We would also like to recognize the Inuit of Nunasivut and Nunatuvut, an Innu of Nitassinan, and their ancestors as the original people of Labrador. We strive for respectful relationships with all the peoples for this province as we search for collective healing and true reconciliation and honor this beautiful land together. We finally encourage everyone to reflect on these lands from where you are located and the Indigenous peoples for whom these lands are traditional territory. Again, welcome everyone, and I would like to introduce again Dr. Stephanie Colombo from Dalhousie University, who will be talking to us about farmed salmonids in Canada. Go ahead, Stephanie.
Hello, everyone, and thanks for joining in this afternoon, and thank you for inviting me. I'll go ahead and share my screen. Okay. So thanks everyone for joining in. My name's Stephanie Colombo. I'm an assistant professor and Canada Research Chair in Aquaculture Nutrition. I'm at Dalhousie University, Faculty of Agriculture, and we are on the agricultural campus. So for the next 40 minutes or so, my plan was kind of to give you an overview of the research that we do in my lab, talk to you about our research goals, where we're thinking of the future, and giving you some examples of the research we've been doing in the past couple of years and what we have planned for this year. So most of the research we do in my lab is on Atlantic salmon and rainbow trout. That's just given the region that's has the most industry focus and research relevance for us. So the title of my talk is Farmed Salmonid Nutrition in Canada: New Solutions, Adaptations, and Ideas. So I'm sure it's no surprise to anyone that's joined this seminar that our fish consumption has increased over the past several years. So in fact, our food fish consumption has increased by about 122% in the past 3 decades. And that's really not due to an increase in wild catch production, because the global fisheries production during this time has only increased by about 14%. So where are we getting our fish from? Aquaculture. So aquaculture has grown over 500% in the past 3 decades. And by the way, these statistics are from the latest State of the Fisheries and Aquaculture report from the Food and Agricultural Organization of the UN. They come out with this report every 2 years, so this is from their latest data. So again, this graph is from that report, and you can see from about 1990 that aquaculture has grown exponentially to the point now where it's supplying about 50% of our seafood. So in the next 10 years or so, we're going to be approaching 9 billion people on Earth. Over 230 million metric tons of seafood will be required. Right now, we're producing about 180 million metric tons of seafood. And 60% of our seafood is going to come from aquaculture. So if that wasn't enough to convince you that aquaculture is a very important part of our future, I'm not sure what else will. And so Canada has a huge role to play, not only in sustaining ourselves with regard to food security, but supplying food globally. Of course, with using our ocean resources sustainably, and really that's where the challenge lies, and that's where my lab's research interest lies, is producing safe, sustainable farm seafood by using our ocean resources in a sustainable way. So I call our lab the Aquaculture Nutrition Lab. Like I said, we're at Dal Agricultural Campus. I know some of you that are here today have been there lots of times, but we're located in Truro, which is about an hour outside of the main campus in Halifax. So our kind of guiding goals, our guiding strategy is to improve aquaculture sustainability and production through new discoveries and innovations. And we do this by looking specifically at nutrition and improving sustainability in sort of a nutritional lens. And really our goal, and my personal goal, is to contribute toward healthier and sustainable seafood for our future. So for those of you that haven't been to the Dal AC Aquaculture Labs, here's a few pictures. We have the capability to run freshwater and seawater trials. Our freshwaters flow through. We have a local aquifer that supplies us with fresh water. As for our marine systems, we actually truck seawater in from Halifax and we run them through recirculating aquaculture systems, and both work very well. Currently on site, we have a number of different finfish species: Atlantic salmon, brook trout, rainbow trout, Arctic char, and striped bass. We have lots of different invertebrate critters as well, like green crabs and lobster. Once we kind of do our finfish trials, like you can see here, we use other labs in the facility. We have associated labs like nutrition and product quality. And these are a couple of older pictures when we could be in person in the lab of some of our grad students. So I want to talk about three kind of main research themes that we work on in my lab. And I'm choosing these research themes because I thought it would be most interesting for this talk today. So looking at sustainable feed resources for salmon, impacts of climate change on salmon nutrition and metabolism, and the nutritional quality of farmed salmon. So again, we focus on rainbow trout and salmon just because of the regional focus. So in terms of nutritional resources, when I say nutritional resources, I mean anything in the diet that can supply protein, fat, any nutrient. And we really focus on replacing fish meal and fish oil. So these are ingredients that are sourced from wild capture fisheries. So of course, it doesn't make sense to be harvesting wild fish to then feed to farmed fish for a sustainability issue and also an economic issue. So aquaculture is heavily interested in not using fish meal and fish oil at all. So that's one of the things that we're focused on in my lab. A few examples of what we're working on, and I'll show some more specific examples coming up, including transgenic camelina oil, microbial and microalgae biomass and oils, for example, schizochytrium and pavlova. We're also working on black soldier fly meal, as well as hemp byproduct proteins and oils. As for functional ingredients and functional feeds, we're looking at hydrolysis lignin and Hematococcus pluvialis. I'll talk about that a little bit later. So as for the first topic, camelina, I actually did my PhD project on the natural wild type camelina. And this was part of a quite large project that was among OSC. So Chris Parrish and Matt Rise were leading this part of the project, and at Dal AC and researchers in Saskatchewan. And there's interest in looking at camelina for a couple of reasons. One, agronomically speaking, it's a very robust crop. So the idea was that we could even grow it in Atlantic Canada, where some of the soil isn't the most fertile for growing, various crops. But there's a lot of potential in growing camelina. The reason that the aquaculture side was interested in camelina is because it has very high level of omega-3 fatty acids. And so potentially that would be a fish oil replacement in diets for salmon and rainbow trout and Atlantic cod. The problem with wild type or natural camelina is that it doesn't have the same profile, fatty acid profile as fish oil. So it's actually missing eicosapentaenoic acid and docosahexaenoic acid, which are the long chain omega-3 fatty acids, which are really the physiologically essential fatty acids for salmon. So we published a lot of interesting and positive research on the use of the natural wild type camelina oil. And even today, there's a lot of interest around commercially using camelina oil in diets for salmon. But so this transgenic camelina is kind of the next generation camelina. A group at Rothamsted Research in the UK has been working on a transgenic variety of camelina for the past 20 years or so. And in the genome of camelina, they've actually included transgenes from microalgae and fungus so that the resulting oil produces a fatty acid profile that is very similar to fish oil. So it does in fact contain EPA and DHA at levels similar to fish oil. That's very different to the natural type of camelina oil, which doesn't produce EPA and DHA. And in fact, no terrestrial plants produce EPA and DHA at all. That's why this variety of camelina is very interesting. So the group at Rothamsted, they collaborated with other researchers in testing this oil for Atlantic salmon and sea bream, but they hadn't yet evaluated this in a freshwater, a species that's grown entirely in freshwater like rainbow trout. And obviously rainbow trout has a lot of industry relevance in both Canada and the US. So we tested this oil in diets for rainbow trout. We had a fish oil control diet. We included a low-level camelina oil diet, which really was just to supply just the bare minimum of EPA and DHA, and then a high-level camelina oil diet. And we raised the rainbow trout for about 3 months, and we looked at their growth, the fatty acid content of various tissues, and we also looked at the compound-specific stable isotope analysis, or the isotopic signature of DHA in the diet and the muscle, as well as a sensory analysis. So this project was funded by Weston's or Loblaws, and graduate student Angelisa Osmond worked on this project, and it's currently under its second revision, or a revision was requested in World Aquaculture Society journal. So hopefully that will come through soon. What we found was pretty interesting. The rainbow trout that were fed the camelina oil diets actually had significantly higher weight gain than the rainbow trout that were fed the fish oil diet. So the feed intake among the groups was similar, but that ended up resulting in a 13% lower feed conversion ratio in the trout that were fed the high camelina oil diet compared to the fish oil groups. And I think the reason for this is that the phospholipid levels in the camelina oil diet were higher, or in camelina, the transgenic camelina oil, were higher than that of fish oil. So I think that some of the fatty acids that are esterified into the phospholipid were more bioavailable to the fish, which resulted in a higher weight gain. Looking at the fatty acid profile, or at least some of the select fatty acids that were stored in the muscle tissue, I'm really interested in EPA and DHA. So supplying rainbow trout with a diet that contains fish oil compared to the transgenic camelina oil as a full replacement really had no difference in the muscle storage of DHA and EPA, which is important for the fish, but also important for us that would be eating the rainbow trout fillets. But we actually wanted to confirm that specifically this group here that were fed the high camelina oil diet, were they storing the DHA that came from the transgenic camelina oil, or was that DHA selectively retained from the fish meal? So there's only a couple of ways of really confirming this. So we actually did a compound-specific stable isotope analysis to look at the isotopic signature of DHA in trout that were fed the camelina oil diet compared to the fish oil diet. And the reason we were able to do this is that the isotopic signature of DHA in the camelina oil, you can see here, is very different than the signature of the DHA in fish oil. And that has to do with the origin where the carbon is coming from, marine environment versus terrestrial environment. So we can distinguish the difference in the carbon from DHA from these two sources. So the oil is different, the diets are different, the DHA and the diets are different. And what we did find is a significant difference in the isotopic signature of fish that were fed the high camelina oil diet compared to the fish oil diet, meaning that we could confirm that the DHA that's stored from the camelina oil was stored from the camelina oil, not from the fish meal that we provided in the diet. So that was kind of a neat analysis that we did to be able to confirm that, yes, rainbow trout are able to digest and absorb DHA and are storing it in the muscle tissue. So that's kind of a unique example of a terrestrial oil that is a pretty good fit to replace fish oil. For the past 10 or 20 years, commercial feeds for salmon have really decreased the level of fish meal and fish oil in the diet by using various terrestrial plant ingredients. And apart from the transgenic camelina, most of the terrestrial oils and plant proteins are not really that great for carnivorous fish like salmon. For one, they don't contain EPA and DHA in the oil. And also the plant meal itself contains some compounds that are useful for the plant as a defense mechanism, but not useful for the fish. So it actually prevents some of their digestion and absorption of the nutrients. And of course, these are called anti-nutritional factors. So there's been really an evolution that even I've seen in the kind of short time I've been involved in aquaculture since about 2006, is this, replacing fish meal and fish oil with terrestrial ingredients, but now replacing those terrestrial ingredients with new ingredients that are a better nutritional fit for salmon and other carnivorous species. And what's really neat about these new ingredients is not only is the nutritional profile better for the fish, but it also uses byproducts from other industries that normally would have been waste. So it's kind of a win-win situation and a lot more sustainable. So there's a couple of examples that we're working on my lab. And another thing that's really neat is that these ingredients are sourced in Atlantic, Canada. So we, produce them right here. So the first example I'll talk about is microbial and microalgae biomass and extracted oils. We're working on a project right now with Chris Parrish and Matt Rise and Sean Tibbetts at NRC, looking at various species of microbes and microalgae as part of the Ocean Frontiers Institute project. And we've had some really interesting results, which I'll talk about in a second. But what's great about the microbial and microalgae ingredients is that the carbon feedstock that they grow these microbes on is also from industry waste. So it would have otherwise been waste, and now you're upcycling and making better ingredients that could be used for aquaculture. Some examples of the waste is spent brewery grain and pulp and paper byproducts. So it's a source of carbon that you can then convert into something useful. So I have a number of different projects in this area. And so there's obviously a lot of interest in these microbial sources. Some of the partners we have on these projects are Mara Renewables, of course, the National Research Council, and Domtar, which is a very large pulp and paper company. The other example I wanted to highlight, and we're just getting started with this project right now, are black soldier fly larvae meal. And you can see this is a picture of the larvae themselves. And another, really interesting thing, the feedstock that the larvae are grown on are also from industry waste. So food and vegetable processing waste, for example. I know the company we're working with, they are using some of the waste from a broccoli plant in Halifax. They also use spent brewery grains and are also interested in using seaweed and fish processing waste. So this is a recent project that we're just getting started with this year. It's funded by the Atlantic Fisheries Fund. And we're working with Oberland AgriScience, who produces the black soldier flies, and also Sustainable Blue, which is a land-based salmon farm in Nova Scotia. So about the microbial oil, and this is the actual picture of the oil that we use, that really nice red color. So we're using the oil from Mara Renewables. They're located in Dartmouth. They've developed a novel strain which originated from the Bay of Fundy. It's grown heterotrophically. What's really cool about this oil is it has a unique fatty acid profile. It's very high in DHA. 40% of the total fatty acids are DHA. It's kind of unique because it doesn't produce much EPA at all, and also produces a pretty high level of saturated fatty acids. But really, the most unique and interesting thing is the level of DHA that it produces. So we've tested the oil in diets for salmon. That was part of the Ocean Frontiers project, and then also with trout, which was part of the Loblaws project. In general, from both species, what we found is that there's really no impact on growth performance at all when comparing salmon or trout that were fed a diet that all of the lipid came from the microbial oil, compared to the control that had just fish oil in it. The other interesting thing is that the resulting muscle tissue, so the fillets, were very high in DHA. In fact, they stored more DHA than fish that were fed the fish oil control, which from a consumer perspective is really interesting because you're actually getting more omega-3, but you're not getting EPA. So some of the implications of that we're not really sure about, but in our experiments, we didn't see any impact on growth or health of the fish. So the fact that they didn't get any dietary EPA didn't seem to be a problem, but probably some more work needs to get done in that area to look at that. You could also mix oils from different species of microbes so that you get a good balance of EPA and DHA. And we published that study just recently. Min Min Wei, who's a grad student in my lab, she was the first author, and Chris was also involved. Nigel did some of the fatty acid work, and Jeanette did some of the fatty acid work as well. So thanks to the team for that. It was a really nice study. We're also looking at microbial biomass. So the same species, Schizochytrium, if you imagine the whole cells are harvested and then freeze dried, and you end up with a meal that looks like this. And so this particular strain of Schizochytrium was grown using feedstock from cellulosic sugars from pulp and paper industry. The company I was working with is Domtar. And so we did a classic digestibility design, which included the diet itself included 70% of a reference diet and 30% of the test ingredient, which was Schizochytrium. And we tested this for Atlantic salmon. So in the first graph here, you can tell that the dry matter digestibility, or the total digestibility of the diet, was not different between the reference in orange and then the test diet in gray. Protein digestibility was very similar between the test diets and also the ingredient itself, the Schizochytrium. Now, lipid actually was, you can see it kind of decreases. It's lower in the test diet and quite low from the Schizochytrium ingredient itself. And that's kind of expected, especially with using this kind of design with 30% Schizochytrium. That biomass contains a very high amount of fat. It contains 60% fat. So when you include that in the diet at 30%, the salmon were getting probably too much fat than they could handle or could digest. So we see a decrease in digestibility. But we know that the digestibility of the DHA in that fat was highly digestible. And so we think that some of the reason why the total digestibility is lower in the Schizochytrium is actually due to the saturated fatty acids and not the polyunsaturated fatty acids. And we do also have the data here on the fatty acid digestibility. And sure enough, the saturated fatty acids were not very well digested. That's kind of typical from salmon and cold water. We know that they don't digest saturated fatty acids as well as polyunsaturated fatty acids. But the really important thing is that the DHA was very well digested in the test diet and the Schizochytrium meal. And the omega-3 and omega-6 fatty acids were both very well digested. So there's a lot of promise in using this ingredient. The practicality of using 30% of this ingredient in the diet is probably, you wouldn't see that in a commercial diet just because the lipid is so high and you have this nice balance of protein and lipid. And also practically speaking of extruding these diets, because the lipid is so high, it's really difficult to extrude. You really should have all of your fat at the end and coat it as an oil. So that's why we just finished a growth study that we looked at graded levels of 0, 5, 10, 15, 20, and 25% inclusion of the biomass. And so this work was done and published just recently by grad student Bailey Hart, and this is part of her master's project, and currently she's working on writing up the data from the growth study. So it's about, ingredients that could potentially replace fish meal and fish oil. We're also looking at functional ingredients. So functional ingredients, they don't really provide a nutritional source in the diet, but they provide other benefits, like they can improve pellet quality or have a beneficial impact on fish health or growth or product quality. Some examples that you would know are pellet binders, pigments, prebiotics. So we're working on a couple of these functional ingredients in my lab. Last year, we just finished a study on an ingredient called hydrolysis lignin. Now, lignin has been used as a pellet binder before in salmon feeds, but hydrolysis lignin is a little bit different. It's made in a little bit different way and it has other properties. And so what we found is that at 3% inclusion, it actually improved the weight gain of salmon by about 16% compared to the control, which is actually quite a bit. And part of the reason for this is that it modulated the gut microbiome and it improved pellet durability. So it does have some functionality as a prebiotic and also as a pellet binder. And we published that study last year in Aquaculture Nutrition, and Ziyu Zhang, who's a grad student on my lab, helped work on that study. The other functional ingredient we're working on is Haematococcus pluvialis, and I'll talk about that in a second. Both of these examples of the functional ingredients are examples of upcycled ingredients. So the hydrolysis lignin is actually a waste byproduct from the pulp and paper industry. And the Haematococcus is actually grown from feedstock from byproduct from the pulp and paper industry. So both are good examples of sustainable resources. So we were working on Haematococcus pluvialis. What's really interesting about this species of microalgae is that it contains a very high amount of astaxanthin. And astaxanthin is important to provide in the diets for salmonids because it lays down pigment in the flesh. And so we get to eat salmon that has that beautiful red and orange color that we like to see. Now, supplying synthetic astaxanthin in the diet is very expensive, but obviously it's necessary. So the industry really is looking for natural, sustainable, and cost-effective alternatives that they don't have to use expensive synthetic astaxanthin. So we conducted a study with rainbow trout, and you can see in this first graph, we looked at colorimeter readings, so the L, A, and B scores, which are measures of lightness to darkness and redness to yellowness and blue to green. So it's a measure of color. And then we also looked at the classic SAMOFAN. So we have measurements from week 0 before they started the diets, a diet that didn't have any astaxanthin in it, a diet with synthetic astaxanthin, and the Haematococcus pluvialis astaxanthin. And the two treatments I'm really interested in are these two, the synthetic and the Haematococcus. And you can see for each of those comparisons, there was no difference in any of the colorimeter scores or the SAMOFAN between including synthetic or Haematococcus astaxanthin. Of course, we want to kind of back that up with measuring the actual astaxanthin content that was in the fillets. And obviously, compared to not supplying any astaxanthin, the synthetic and Haematococcus were both significantly higher in terms of total astaxanthin. But importantly, there was no difference between supplying the synthetic and the Haematococcus, showing that you don't need to use synthetic astaxanthin. You can use this the Haematococcus astaxanthin. And then also in terms of product quality, the total lipid and total protein of the fillets wasn't different between any of the groups. So typically Haematococcus is quite difficult for fish to digest because it has a really tough cell wall. But the industry partner that we're working with, Kwelne Agrisystems in Hawaii, they produced a novel product of Hematococcus that has a very weak cell wall. And so that obviously shows some really promising results in regards to the ability for fish to digest and use the astaxanthin. Of course, astaxanthin also has other properties other than pigment. It's shown to have some protective properties as well, partly because it is also known as an antioxidant. So we have a new exciting project coming up to test the wound healing capacity and the pigment effectiveness of Hematococcus. And we're working with Mark Fast at UPEI, Aqua Bounty, and Sean Tibbetts at NRC as part of the OFI One Ocean Health project. And I'll be looking for a PhD student to start in May on this project pretty soon. So kind of moving on to the next theme, I'm interested in the impact of climate change on salmon nutrition and metabolism. And I started working in this area when I was doing a postdoc at Ryerson University. And it kind of started with the idea and realizing that a lot of single studies on algae were showing that, you know, at a lower temperature, algae produce lots of polyunsaturated fatty acids like DHA and EPA, but at a higher temperature, they produce less EPA and DHA. So what does that mean on kind of a global scale? So what we found is that an increase of 2.5 degrees by the year 2100, so about 75 years from now, can actually result in lower production of EPA and DHA due to the increase in temperature. And so the reason for this is why DHA is synthesized and why it's used by primary producers in aquatic ecosystems. So at really low temperatures, the algae tend to produce more DHA to help with fluidity of the membrane to basically help the cell from freezing up. But at a warmer temperature, they don't need to produce as much DHA because the membrane is already quite fluid. And so they actually need some more structural integrity in the cell membrane. So not as much DHA needs to be produced. So if you think about that in the context of climate change, where we're seeing global increases in water temperature in both marine and freshwater ecosystems, we collected a very large data set and just applied a very small temperature increase, which is considered a very conservative climate change model. And what we found is that global DHA production by phytoplankton, you know, bottom of the food chain, was shown to decrease by 30% in the next 75 years, which is actually pretty interesting. But what is that? How does that transfer to higher trophic levels in the aquatic ecosystem? And how does that transfer to fish? Because that's where we're getting our DHA. So how is the global availability of DHA for human consumers going to change as a result of increasing temperatures because of climate change? And so we have published a couple of these studies so far, and I'll show you some of what we did. So in terms of looking at the production of DHA by fish and what's available to humans, we first wanted to know what's the base case, like how much DHA is produced on an annual basis right now. And what you're seeing here is kind of a color-coded map, and it's divided by fishing zone of how much DHA is produced by fish on an annual basis. So first we had to know how much fish is harvested, is grown or harvested in each fishing zone. So we got that data from the FAO and the Sea Around Us project out of UBC. Then we had to know of what's harvested, what is edible portions. Of the edible portion, how much of that is fat, and of that fat, how much is DHA? And that's how we came up with kind of the base case of what we produce annually, or what fish produce annually for human consumers in terms of DHA. And so the base case scenario is that we currently harvest both from farmed and wild caught fish is about 370,000 tons of DHA per year. That corroborates with some other research that some other people have done, like out of Scotland, for example, that we're in the right ballpark. So what we found is that applying that model, what we did with phytoplankton, it could reduce DHA production by about 58% on average. Just assuming that whatever is produced less by algae, that would translate into fish and transfer through the food web. Of course, it depends on geographic region. We did find that freshwater ecosystems in temperate climates will be more susceptible to decreases in DHA. And so this 58% reduction equates to about a decrease to about 140,000 tons of DHA based on the most conservative IPCC climate change model. So this does really highlight aquaculture's role in providing a more consistent source of DHA through cultured fish. So if salmon are fed diets that don't have fish oil anymore, we could supply the fish with a more consistent supply of schizochrysarium oil, for example, so that we know how much DHA we're going to provide them. That's still a work in progress, but it does show the potential that aquaculture has in terms of providing a consistent source of DHA for consumers. Of course, like with any model, you're going to have model assumptions. And the biggest assumption that we made is that what we see in phytoplankton is going to transfer up the food web to fish. That's a pretty big assumption, but it's also a reasonable assumption because we know that DHA is selectively retained in aquatic food web. So what we see at the bottom, we're probably going to see at the top because DHA is making its way up there. But still, it was an assumption that I wanted to test in the lab. And what I wanted to know is, does higher temperature and coupled with a lower dietary DHA impact fish metabolism in real life? Because here we were just looking at, with the model, we were just looking at the storage of DHA. We didn't look at the impact of metabolism on increasing temperatures and also decreasing an essential fatty acid. So that's what I wanted to do in the lab. And I wanted to answer the question, what's the impact on health, growth, fatty acid metabolism, and omega-3 synthesis when fish are faced with a situation where there's increasing temperature and lower DHA. So we did a multifactorial trial. We did this last year. Even during COVID, we managed to get this experiment done, which I'm really proud about. We had two diets, a low DHA and a high DHA, and each of those diets were, the fish in those treatments were reared at two temperatures, 12 degrees, which is like the control, and then at 16 degrees, which is more of like a climate change scenario. So we raised Atlantic salmon in seawater for about 4 months. And of course, this has implications on pen-raised salmon, but also has implications on wild salmon populations as well. What we found, this is some preliminary growth data. What we found was actually pretty interesting and not what I was expecting exactly. So this is the weight gain, and we have our 4 treatments here. High DHA at the lower temperature and low DHA at the lower temperature, and then high DHA at the high temperature, low DHA at the high temperature. This is the treatment I was really interested in because this is like the climate change scenario group. And they actually grew more than fish that were fed the high DHA lower temperature. Now, because of the temperature, I kind of would have expected that this group might have grown more, but I would have expected this group to grow more because not only were they raised at a higher temperature, they were provided with more DHA. So that was kind of interesting to me. And even comparing fish that were raised at the same temperature, even though it's not significant, you can see there's a proportional difference here between these groups. And then looking at feed intake, fish that were fed this climate change scenario treatment, they ate more food than any other treatment. And first I thought this was kind of counterintuitive because I thought if you're given a better diet, then you're going to grow better and you're probably going to eat more, but not necessarily. And I think maybe the reason for this is because that they didn't have a lot of DHA in their diet, so they had to eat more to compensate for that, so they could still get the same amount of DHA. I think that might be why. I'm still working it out. We have some more results to look at. But so salmon grown at the higher temperature fed the low DHA diet grew the most. That's the climate change scenario. They also had a higher visceral somatic index, meaning that those fish stored more fat around their organs than then layering that fat into their whole bodies. So that's interesting. They also ate more food. So what does that mean for growing salmon in pens in the future where the temperature is going to increase? And if we're using fish oil that has less DHA, or we're just feeding diets that really don't have much DHA at all in them? And then what does that mean for wild salmon populations? Are they going to be consuming more food? And what about that prey availability? So there's a lot of questions, unanswered questions, and we're still working on the results. So we're doing a fatty acid mass balance kind of inventory with Suzanne Budge at Dow. Jennifer Hall is doing some of the transcriptomics for us. And we're also looking at the enzymatic activity of the enzymes that are involved in synthesizing DHA. And that's being done by colleagues at the University of Toronto. So I'm hoping these results will kind of lead to another experiment, maybe developing some functional feeds that will actually help salmon grow better at higher temperatures, things like that. But I just wanted to share some of those preliminary findings so far. It's really exciting to see. It will be exciting to see the new results that come out. So far I've presented, I think, some challenges in aquaculture like fish health, sustainable feeds, climate change, waste mitigations, that I think will have, there's solutions to each of these with the right scientific innovations. But one of the biggest problems that are faced by the farmers and the aquaculture industry as a whole, particularly in Canada, is social perception or public perception and acceptance. And I know this because I'm a scientific advisor for the Aquaculture Association of Nova Scotia. And I also happen to know this regarding the Newfoundland Industry Association, is that part of their strategic plan, actually the biggest part of their strategic plans for last year and this year, is trying to improve public perception. And so I find that really interesting because of course we're working on science-based solutions, but how can we translate that to the public so they, have better trust in the products that they could be consuming? And so one of the things that I hear a lot is, wild salmon is just better for you, and that people won't eat farm salmon because it's not as good for you as wild salmon. I really do not know where that came from. Where are they getting this information from? I certainly haven't seen anything like that. And really, when I started to dig into the literature, there's not a lot of data out there to show scientifically if there is a difference, nutritionally speaking, between fillets from wild salmon compared to farm salmon. So I just wanted to run this fun little project to look at what's the nutritional composition of different wild salmon and different farm salmon. I also want to know, is organic salmon better, like nutritionally better? Did different environmental certifications have any weight with regards to better nutritional profiles? And I think, honestly, I didn't even have this information at my fingertips. So I think part of the reason for some of the confusion is that the nutritional labeling of fresh seafood, like you'd see at the counter, like you would see for farmed Atlantic salmon, the nutritional labeling is not required. So it kind of leaves the consumer in the dark. So we wanted to run this study and look at the nutritional composition of different types of salmon that are available to everyday Canadian consumers. So I tried to collect as different samples as much as possible that looked at different certifications and different species. And myself and honors student just collected, salmon from Sobeys and Superstore, or it's Dominion in Newfoundland. And also online. And our stipulation was that it had to be Canadian source. So it had to either be caught in Canada or had to be grown in Canada. So we ended up coming up with 6 different types: farmed Atlantic, farmed organic Atlantic, farmed organic Chinook, wild Pacific pink, and wild sockeye. And we analyzed them for a bunch of different nutrients: protein, fat, fatty acids, amino acids, potassium, iron, cholesterol. And I also wanted to look at least in one example of a contaminant. And so I chose to look at mercury because it's the only contaminant that Health Canada has an advisory for consuming seafood for Canadians. And also, I had a budget for this project, so I couldn't really afford to do all the PCBs and other um, organic pollutants. So we chose mercury to focus on. So maybe no surprise to you, but I think this is a surprise to the public that it's really about the species of salmon that makes the biggest difference in the nutritional quality, not whether it was farm raised or wild caught. Even within the group of wild salmon, there were really big nutritional differences. So we published this late last year, and we actually got a little bit of media interest in this, I think, because it's a kind of something topical that not a lot of people know about. So there was an article on CBC, and we did some radio interviews and that kind of thing. But I think there's a lot of space for this in, science providing some answers for consumers that really don't have this information at their fingertips. So this is a principal coordinates analysis. I really love doing these. Chris taught me how to do these. I'm very grateful for that. And now I'm teaching the students in my lab how to do this. So each data point represents the entire nutritional profile from a fillet. And so out here, you see wild Pacific. Down here, you see sockeye and Chinook. Up here, you're seeing farmed organic. You're seeing Atlantic. You're seeing Chinook. And so again, it's really, if it was about farmed and wild, you'd see these two groups like this, but they're kind of spread out. And some of the vectors mean that they are highly correlated with the samples that they're close to. So for this Pacific salmon, you see a lot of the amino acids. There's a lot of moisture content in the Pacific salmon and has pretty high protein content and is very lower in fat. So that's why you see all the protein and amino acids here. With the sockeye and Chinook, we're seeing a lot more of the marine fatty acids like EPA and DHA. And here we're seeing some of the more terrestrial based fatty acids that you would see in terrestrial oils that are supplied in the diet for some of these species. So the most nutritionally dense species were wild sockeye, wild Chinook, organic Atlantic, and farmed Atlantic. Those were the most nutritious. Wild Pacific was the least nutrient dense because it had the highest water content. So every bite you take is more water than anything else. So wild sockeye and wild Chinook, they were the most nutrient dense and they had the most omega-3 content, but they're also the most expensive and they also had the highest mercury content. And there's also sustainable limits to the catch. So we can't be eating wild sockeye every day. Not every Canadian can be eating wild sockeye every day because it's just not enough fish. And also you want to watch your mercury consumption. So farmed Atlantic really did turn out to be the best option for people who are often including salmon in their meals. In terms of the nutritional value, its availability to consumers, had the lowest mercury content and was very low cost. So I think some of these misconceptions regarding wild versus farmed is just because the public doesn't have all this information at their fingertips. We know that traceability and labeling of our food is evolving. So I think it's important that we provide as much information to the public as much as possible. And so maybe this has implications for how we label fresh seafood. It would be nice to be able to put labels like high source of DHA, which is important for brain health or something like that on a farmed salmon product. So I really enjoyed doing that study. It was kind of fun and led to some pretty interesting results. So this is my last slide just to sum up kind of the driving motivation for my lab and team are, how can aquaculture do better? We have a lot to do in the future. So there's so much to work on. It's really about this kind of triangle of ocean health, food security, and sustainability. We want to know how we can use less resources to make better food. How can we rely less on wild fisheries to help improve ocean health, but also so that we can harvest more seafood? How can we improve the resiliency of aquaculture in Atlantic Canada, particularly when we're faced with a changing climate that has, warmer temperatures and salinity and things like this? And really, how can we improve the nutritional value of our farm seafood? So thank you very much for listening. Thank you to the grad students that worked on this, my collaborators, obviously all of the funders that were involved. Here are some of the papers that I was referring to, and I invite you all to check out our lab website at aquaculturenutrition.com. And I'll just put the plug out there that for the One Ocean Health Project, I am looking for a PhD student to start in the spring. And for the Black Soldier Fly Project, I'm looking for a master's or a PhD student. So I will say that. And with that, I'd be happy to take any questions, and thank you for listening.
Thank you very much, Stephanie. If anyone has any questions, please feel free to unmute yourself and ask away, or you can put your question in the chat box.
Hello.
Hello.
Hi. I was just trying to find that last publication that you were talking about. I was wondering how many fish you had in each different group for the wild versus farmed.
Right. So we did duplicates on each of the fillets, but we had 3 fillets per category.
Which you sampled each fillet twice.
Yeah, we just wanted to make sure that there was no variability within one fillet, but we ground up every fillet, so it should have been homogeneous anyway. We just, as a double check, did that in duplicate, but the real number per group was 3 fillets. And so we're planning on actually doing another study a bit bigger, looking at like Scottish farm salmon compared to Norwegian farm salmon compared to Chilean, and then also rerunning what we did, but on a larger scale with more samples.
Very cool. Thank you.
Yeah.
Hi, Stephanie. Very good
Hi,
presentation as usual. You're doing some really good stuff. It's totally excellent. I'm surprised perhaps to see you working with transgenics, but there we go. And the other thing that was quite interesting to me was the hydrolysis lignin. It never occurred to me that you could actually use that as a functional ingredient. I happen to have a student who's working on lignin from environmental science perspective. So that was actually very cool. Cool. I wondered about your high DHA, high temperature. Did you look at oxidation?
In terms of what? No, I didn't look at any oxidation, but what oxidation are you referring to?
Well, oxidation would be if you have, first of all, you would reduce the amount of DHA that would be available, but also you would produce certain oxidative products that would be anti-nutritionals, but could also be more toxic. So I just wondered if the oxidation status of a high DHA diet at a higher temperature might be an issue.
I don't think that we would have had any oxidation in the diets themselves. I wasn't sure if you meant oxidation like later in terms of the fish metabolism or the diets, but I don't expect that there was any oxidation, but maybe it's something to check. So
one thing, an obvious one is if the diet smells, right? So
yeah, I mean, it was stored completely at -20 the whole time during the 4 months. We made the diets immediately before we started, so I really wouldn't expect that was an issue. And even now, I just took samples of the diet from the -20 freezer and put them in the -80, and they don't smell at all. So this was just a few months ago. So I don't think that was part of the reason of why we saw what we did.
And you got lots of antioxidants in the diet anyway, right?
And the diet has antioxidants anyways from our special premix. So yeah.
Right. So the other thing is that I thought very nice, the wild versus farmed study was it's a shame you couldn't do the, I guess the various pollutants that would be hydrophobic.
Yeah.
Because I mean, we did a paper exercise years ago, it would be out of date now, where the whole thing of feeding lower trophic level and cleaner products to aquaculture species should have a much lower body burden than you would get from wild species.
Yeah. I know. Well, we did see that for just mercury, but it would have been nice to have a little bit more power there. Actually, a new study was just published and they cited our study, and they did just that. They didn't look at really the nutritional content as much. They looked more at the contaminants, and what they found was exactly what you said.
Right.
Yeah. So that's a brand new study that just came out maybe last month or something.
Excellent. I will look for that.
Maybe
will
if
look
I
at
have
some
a
of your-
bigger budget, I would do all of those, but it's very expensive to do because I wanted to do that, but it's not cheap.
Yeah, and there's all kinds of protocols that you have to worry about in terms of how you sample and don't contaminate and stuff like that.
Right.
So,
Yeah, good point.
Yeah, it is tricky. Anyway, well done.
Thanks.
Hi, Stephanie. Good to see
Hey,
you.
Dwayne. Good to
Hey.
see you too.
Fantastic.
You're right on the salmon farm, I see.
Yeah, exactly. Right here, sitting down outdoors. Yeah. Great work. You know, it's so nice to see everything you're doing there and so on. So I'm just curious, really enjoyed all the results you presented, of course. But the one about the climate change really got me thinking, right? So here in BC, we get warm water events. We're up to like 18 degrees sometimes and even hitting 20. So just curious, have you tried any other higher temperatures, or is that some plans for down the road or anything like that?
Well, honestly, originally when I proposed to do this experiment to animal care, I had 18 degrees and they're like, nope, you're not doing 18 degrees. I was like, nuts. But because I know that's really where it's going. Now we would see spikes like here in Nova Scotia up to past 20. But those are spikes, that's not like a constant level. And for this experiment, I was a little bit concerned at running our RAS, because this is in RAS, at a consistent 18 degrees. I don't know if we could keep up with it. And so just because of all of those things, I just compared this 4-degree difference, even though, you know, in my mind, I know the 18 is probably more appropriate looking in the next few years. Actually, even now at some times in the year, late August, September, even for, especially on the West Coast.
Yeah. Yeah, awesome. No, good to hear. But again, yeah, great stuff. Keep it up.
Yeah, thanks, Dwayne. Good to see you.
You do.
It's good to see so many familiar faces in the participants list.
Is there anybody else? Okay, well, with that, we will end the seminar. Thank you so much, Dr. Colombo, for coming and speaking. And thank you, everybody, for tuning in. And we'll see you all next week.
Thanks so much for inviting me.
Yeah.